Dr. Leslie, are you ready?
Good morning,
Good morning,
everyone.
everyone.
Oh, I have an echo.
Okay, maybe I had need to.
Good morning, everyone. Welcome to the second half of this session of the 40th APAMS. So this session is for biotechnology, and we have three speakers. Our first speaker is from the University of Santo Tomas. She is Dr. Donna Papa, an assistant professor in the Department of Biological Sciences, College of Science, University of Santo Tomas. She heads the UST Bacteriophage Ecology, Aquaculture, Therapy, and Systematics, or BEATS, research group. She took her master's in microbiology and PhD in biological sciences from UST and was awarded a research internship at Université Laval in Quebec, Canada. This was supported by the Canada Research Chairs and DOST BCDA PCIRD. She is a diplomate of the Philippine Academy of Microbiology, an academic committee member of the International Phage Research Center in Nanjing, China, and is the corresponding secretary of the Philippine Society for Microbiology. So I will now give the screen to Dr. Papa to talk about bacteriophages.
Thank you so much, Mom Les, for that introduction. So let me just share my screen. Okay, so good morning, everyone. Thank you, PAASE, for this invitation to present some of our works in UST. So this morning, I will be presenting to you the prospective bacteriophage-based researches for application in medical, food, and aquaculture industry. So I will be showing you some of the works that has been done outside the country, and even some of the things that me and my students have been doing in our laboratory for a couple of years now. So we all know that in 1928, Alexander Fleming accidentally discovered penicillin. But because of the superbugs that has been rising right now, there's now this scary threat to modern medicine of this antibiotics resistance. This is one of a major problem right now, not only in medicine, but also in the food industry and also in the aquaculture and agriculture. So, phage biologists propose this re-emerging method, which is the use of bacterial viruses or bacteriophages, which are very host specific. They infect only bacterial cells. This is proposed to be an alternate to antibiotics for treatment. The bacteriophages are the most abundant entities on Earth. They are found anywhere and everywhere bacterial organisms are found. That is why they are a very good um, uh, method to solve antibiotic resistance, to treat antibiotic resistance. They are usually found and usually used in different aspects in society. In the food industry, they are used as therapy, biosanitation, biocontrol, and biopreservation for final products by consumers. Just to give you an example, there is one company in the US, the Intralytics Incorporated, which actually has a lot of products for the food industry. One product, the Listex, which was licensed in 2006 by the FDA, is the very first phage product in the food industry to receive generally regarded as safe by the FDA. It is used in meat, cheese, fish, and other products like RTEs. Here's a list of some companies worldwide that are producing phages for the food industry. So in Canada, Netherlands, and the US. In the aquaculture industry, the improper use and improper administration of antimicrobials has already rendered also antibiotic resistance. That is why there are a lot of studies being done already directed to these pathogens examples, Pseudomonas, Vibrio, Aeromonas, etc., and on different animal models. Lecumanon et al. In 2016 proposed a method on how bacteriophages can also be applied in the aquaculture. There are not a lot of products in phages for aquaculture yet, but a product that was produced by a Scottish company won an award in 2018 because it addresses the major challenges of seafood production, like food safety, fighting antibiotic resistance, and feed efficiency. So that is one of very promising product prospect for bacteriophages. Now, for I think my favorite, or this is one of the things that phages are really well known. Um, Phage therapy. So I'd like to introduce to you the George Eliava Institute in Tbilisi in Georgia, which is the top institution in the world that actually accepts patients for bacteriophage treatment. So if you can see here, they have a data from 2007 to 2013, wherein a lot of patients from all over the world are actually going there for treatment, and also and also uh, they are also they have different divisions like therapy center, you have the bio preparations and the biopharmacy, wherein you can just go there and get a prepared bacteriophage already, or you can bring a patient and have a customized phage preparation. So to give you an example, here is a treatment being done. It was done in Eliava Institute a couple of years back of a minor explosive injury, wherein a patient was not responding to antibiotics anymore. Um, here is now the patient. Here is now the patient. Uh, a few years back, a few months after the treatment, and then also bacteriophages can also be used for diabetic wound healing. So you can see here the treatment of phages a few months. This is a patient that was not responding anymore to antibiotics also. Okay, so from here there are a lot of companies because of the results that I have just shown you. Um, These are the different companies. You can see them in phage.org, in Bacteriophage Ecology Group, .com. So there are actually a lot of companies already that are investing millions of dollars just to produce phages. Okay, so you have their phage therapy research. You have their if you have a phage product wherein you want it to be mass produced or manufactured, then you can go to these companies. And also, I already mentioned Eliava. And then these are some phage products that are actually actively being used today. Um, I'm not gonna enumerate all of them, but I'm just gonna highlight one product, the first one here, which is the Phage Bioderm. It is a skin patch wherein it has a cocktail of phages. Cocktail meaning it has two or more bacteriophages like E. coli, Pseudomonas, or Staphylococcus that are put in the patch, and it can be applied on a patient. Okay. So these are just a few of the products. So that was a glimpse of what is being done outside the country. Now I'm going to show you some of the work that we have done in UST with my students, my former students, for a couple of years now. So if you can see later on, no, our project are still actually in the infancy, not as advanced as the others, but at least we have jump-started the work on phage research in the Philippines. And I will not be too technical about the papers that I'll be presenting to you. I'm just going to highlight the phages and at least one contribution of the study that we have done. So in our laboratory, we don't buy phages, but we isolate our own bacteriophage. We characterize them morphologically, and also we check their stability to physical and chemical agents, and we check their host range. We produce mutants so that we can check the activity of the bacteriophages just in case there's a mutation. And then right now we're also we have ongoing a molecular characterization of our phages. It's very important to really characterize and get to know your phages before you bring them to war, against these bacterial organisms. So so these are my students, former students, and students right now. the projects are the ones that are represented to be presented here. So on food industry, we check on bacteriophage-based biocontrol of salmonella in selected RTEs or ready-to-eat foods. So we have here isolated 3 bacteriophages from sewage samples, and then we tested them on pepper pastrami, chicken ham, bologna, bean sprouts, lettuce, in 2 different storage temperatures, which is 4 degrees and 25 degrees. We also did this before in dressed chicken. So we have similar results wherein the cocktail of phages rendered significant decrease of bacterial load in these RTE foods. And then also, a biosensor was developed using a phage bioreceptor to detect salmonella. This is in collaboration with the chemistry department of my university. So the biosensor that was produced is more time efficient other than if you compare it to the other conventional methods. Of course, conventional methods are highly specific. However, they can be time consuming and not usually how do you say this applicable on site. That is why this biosensor can address that concern for on site diagnosis. So for this one, we looked at, for aquaculture, we look at the treatment of an infection, which is motile Aeromonas septicemia caused by Aeromonas hydrophila. We tested this on Orechromis niloticus or tilapia. So we isolated 2 bacteriophages, phage B614 and phage UP87. So after blood analysis of specimens, cocktail therapy showed fastest rate of bactericidal activity compared to a single phage treatment and to tetracycline, which is the antibiotics that we compared it to. So it showed better, fastest bactericidal activity. And then to further that work in aquaculture, we look at I isolated more bacteriophages from Lake Taal and Montreal in Canada. We also use these phages for tilapia, for ornamental fishes like koi and goldfish, but I'm not going to show you all the data here. I'm just going to show you just a few pictures of the treated and the untreated tilapia that where we apply the bacteriophages. So comparing them, you can see here the treated with bacteriophage, and then you can see here the untreated, wherein you can see hemorrhage, fin rot, and scale shedding, and the organs are actually almost liquefied already. So these are promising results, both even in our ornamental fishes. And then for phage therapy, you should always see the phage delivery, or how do you deliver the bacteriophages. A lot of publications usually use intraperitoneal route, oral route, or phage impregnated feeds, and then also bath route or the immersion. So we try to compare all these methods being used and being published, and then we looked at the activity of our phages. Blood samples and kidney samples were checked, and then bacterial load decreased, and bacteriophage concentration increased alongside that. Notably, the IP route is more effective. Unfortunately, of course, it is more invasive. The efficacy of the oral route is actually depending on the fish intake of the feeds. And then the bath route was least effective, but take note that these 3 methods significantly reduced bacterial load. It's just that comparing the 3 of them, the bath route was least effective, but it can be proposed for prophylaxis of, let's say, for example, ornamental fishes. Okay, so, and then for therapy, we looked at burn wound infections. So we isolated the bacteriophage from sewage sample, and we looked at, this is bacteriophage MBO8. We placed it in hydrogel, used it for a single topical application, wherein phage MBO8 was more effective in one single application compared to the multiple application of antibiotics, colistin and gentamicin, which was used in the study, in treating the burn wound infection caused by a clinical strain of Pseudomonas aeruginosa. And then lastly, one of the studies that we have done with my students also, we tested clinical strains and environmental strains for the production of biofilms, because as we know, sometimes with hospital-acquired infections, this is a big problem. Okay, so we isolated bacteriophages. We have here 6 virulent bacteriophages, and we tested them against the biofilm formers, and phage cocktail therapy efficiently eradicated the antibiotic-resistant E. coli and Pseudomonas aeruginosa biofilms that were used in the study. So I just wanted to highlight, letter C. This is a bacteriophage named after my advisor for my master's degree. This is named after Dr. Ireneo J. Dogma Jr., very well-known scientist in the country. So the phage named after Dr. Dogma had the best and most little activity among the phages that we were able to isolate. So that's for our studies. Just a few takeaways. These results, the ones that I presented, make good prospects for further development of bacteriophage products. Aside from phage therapy, we have food and aquaculture industry, very promising. Phage biologists like me, we're not here to replace antibiotics. We're just here to give another proposal, give another option, an alternative treatment, because we know that bacteriophages brings a new hope against our battle against antibiotic resistance. So with that po, maraming salamat. I'd like to thank the following who have supported all our work through the years, and all my students. Maraming salamat po, and good morning po sa inyo.
Thank you very much, Dr. Papa. Just a quick question, because we have an open forum later after the 3 speakers in this second half of the session. Dr. Papa, are there specific applications of phages for certain infectious diseases?
Well, certain applications, yes, ma'am. Aside from the ones that I already mentioned, there are, I think, actually a lot of bacterial infections, even TB, can actually be, phages can be used for them. I'd like to cite one example. We have lung infections like for Klebsiella or Pseudomonas aeruginosa. We actually did a nebulization project wherein we use a nebulization using phages. These are actually being used in El Java also. So we did that, and other applications, you can also use them even for dental caries, even for dog infections, ear infections, because bacteriophages can be used in patches, in sprays, ointments, suppositories, tablets, and also nebulization. So yun po yung other applications for bacteriophages.
Okay, thank you, Dr. Papa. That's interesting to know that specifically the applications for human infectious diseases, because you have mentioned applications on aquaculture as well as other human diseases, but TB, that's good to hear that it has applications on TB and other lung infections. So we'll get back to you later